here, I can talk about all of these. With the red light came on?
Yeah.
Okay. So, there was a mistake on my PowerPoint, and then nobody pointed out. I figured that by myself. All by myself, without your help. How about that? Aren't you proud of me? Okay, the homeostasis, that's the right spelling. Okay. What I had done before, it was hemostasis. Hemostasis. It means when the blood stops. Stasis, as you know, it means stop on your prefix suffix. Is that right? Am I making some sense? Okay. Homeo, it means same. Homeo, same, similar. So when the conditions inside of your body, the definition of homeostasis, when the conditions inside of your body should stay same, it cannot fluctuate too much. Okay, it does change to some extent, but it cannot change too much. If it does change too much, then Houston, you got a problem. What I had before in here, you guys, it was hemostasis. It means when you cut yourself and then the protein factors starts, the blood clotting factors and the other factors start, then that would be hemostasis. Okay, but anyhow, you know what that is. I'm done with it. So last time, how far did we go? Does anybody know how far we went? We went to antibody. We talked about that before. Respiratory system. Is that right?
We just start the respiratory system.
We just stopped at respiratory system, right? Here. Okay. So your respiratory system, guys, if you would, it is for, of course, we moved to the land. And then since we moved to the land, we got rid of our gills, and then we have lungs now. And what happens, your respiratory system is a tree. Think of it as a tree. Reverse it, upside it down, and stuck it down your throat. That's what it is. The trunk of the tree is your trachea. Is that right? That's the trunk of the tree. And then it branches. The branches are bronchioles, and bronchus is the branch, another branch, but for the lab purposes, I want you to know bronchioles. At the end of the bronchioles are the leaves of the tree, which are alveoli. Alveoli is made up of simple squamous epithelial tissue. I hope I'm making some sense. You all know what simple squamous epithelial tissue is. And then, of course, alveoli is surrounded by capillaries to take up oxygen from the alveoli. The capillaries pick up oxygen, and then they give off carbon dioxide, which they took from tissues. They give it into the capillaries, and then it will go back. And then one other quick thing I sometimes mention, that smooth muscle surrounds the bronchioles and bronchus and trachea, and then people who have asthma, these are involuntarily, you do not have control over them for whatever reason. Okay, they contract, and the person has asthma, they cannot breathe, as you know. Okay, so that's pretty much is the histology, anatomy of what I want you to know about nervous system, respiratory system. Not nervous system. Okay, then what happens, you guys, when you are sitting down right now, watching TV at night, when you're asleep, you do not focus, except meditation. When you meditate, that's different. When you're not focusing on your breathing at night, you don't. When you're asleep, is that right? Or you're watching TV, you say, "Oh, Amir said meditate. I'm not going to watch TV. I'm going to meditate." No, I'm not saying that. But what happens? The pressure in the atmosphere, is that right? The pressure in the atmosphere is higher than the pressure in your lungs. So what happens from your physics or chemistry classes? You learn that the pressure goes from high to low. This is lower. So the pressure goes in during inspiration, when you're inspiring. So when the air goes into your lungs, what happens during expiration, the pressure in the lungs are higher than the pressure in the atmosphere. And that's what happens, the air comes out. Okay, so it's automatically being done. I know you might say, oh, I mean, diaphragm is involved, intercostal muscles between the ribs are involved. Yes, whole bunch of stuff are involved. But in simplified version of it, what I would like you to know, you never thought about it, you've never been told that the pressure, difference of the pressure between the atmosphere and the lungs also is involved in your inhalation and exhalation, or inspiration and expiration, or those terminologies. Okay, so that's what happens. And this is the PowerPoint, I hope. Oh, no, okay. So this is what we have in the lab, the bronchioles or bronchus, and these are alveoli. There's alveoli, 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 like a Swiss cheese. And these are simple squamous epithelial tissue. If I have the pointer here, these pointers are microscope pointers. If I have it here, that would be simple squamous. Oh, I mean, that look like more than one layer. No, it is one layer. Okay, thank you. Look at them in the laboratory and we separate them and so on. Okay, again, the difference of the pressure, same slide, yes, I guess. I'm repeating myself. Oh, same slide again. It shows you where the heart is. Your left lung, you have 2 lobes because of this guy, the left side. And the right side, you have 3 lobes. And that's not significant for this class. I don't want you to worry about it. Again, I'm showing you high magnification of what's going on in here. That's what I just talked about it during inhalation and exhalation, inspiration, expiration, what happens, the difference of the pressure. Quickly, let's move on to the next thing I want to say. Okay, the next concept, that what happens, was a lab question, right? The lab discussion question. This is the answer to it. What happens? When you inhale oxygen, when oxygen goes from alveoli, let's just go over this diagram quickly for a minute, and then I hope a lot of things sink in later on. What happens when the oxygen goes into your blood vessels? Specifically, if you want to know your pulmonary vein, which takes the blood into the heart, right? This would be your pulmonary vein. Look at the oxygen level, it's about 100 millimeters, and carbon dioxide about 40 millimeters. Okay, almost same as what's in the lungs. Are you guys following? You don't have to memorize the numbers, but these are what I want you to know. So the pressure of oxygen, the partial pressure of oxygen is greater in the arteries, the vein that is going to the heart, and of course in the lungs. Then blood goes to the heart, we already talked about that. Then the left side of the heart pumps the blood to the rest of the body. When it goes to the rest of the body, that is your systemic arteries. It's systemic, it means all over. So all of the arteries inside of your body has the same oxygen level. Okay? And then it goes to the tissues and, of course, low levels of carbon dioxide. That's why in diagrams, in animals, when they sacrifice them for you guys to dissect and they inject them with special chemicals, you will see the arteries red. Okay? Then what happens in the tissue level, as you learned in biology 1, cellular respiration, what happens, oxygen goes into the cell, and in mitochondria, right? You remember the mitochondria, the footprint, the matrix of mitochondria, the cristae, and so on and so forth, all of the mitochondria structures. It will take that oxygen and break down glucose molecule, and what is the end result of glucose molecule? What is the waste material? Carbon dioxide, right here. So carbon dioxide from the cells of your body, cells in the brain, liver, muscle, bone, whatever cells you have, are given off to the capillaries, and capillaries to the venules, and eventually the venules are going to bring it to inferior vena cava, and superior vena cava goes to the heart. You remember that? And the heart pumps it into the lungs. I hope I'm making some sense. And now look at the level of the oxygen and carbon dioxide. in the veins. The level of carbon dioxide is 46, it was 40 here, so there is more carbon dioxide here in the veins than in the arteries. Am I making some sense, everybody? And then look at the level of oxygen, 40, 100. The level of oxygen is way lower. What happened to that oxygen? To the tissues. Why carbon dioxide level increased from 40 to 46? Again, do not worry about these numbers. Why the level of carbon dioxide increased? Because cellular respiration. The cells are giving off carbon dioxide. And then, of course, it goes up and out, and then we inhale oxygen, and then the cycle goes on and on. And that was a question in the lab. Or if I have my glasses, quickly, let's break out back and forth. Sorry, the question, I have to open up the file. What is this? How come you don't know? Ooh, 5 hours. Okay, so the question I asked for the answer for that one, what is anatomy respiratory? How does respiration occur? What is the difference? Here we go. Question number 25. What is the pressure of oxygen and carbon dioxide in different parts of the body? Okay, what are the differences? Explain. So that's pretty much the answer to that question, and I would like you to know something about it before next Thursday. Okay, so let's move on to the next. Here we go in the subsequent slides. Explain it. I don't expect you to know all of the name of the enzymes here, all of the chemical reactions here. That's for physiology or cell biology when you take it later on. But it's in your notes, and you know it's too much to ask just for this example. Okay, this one example. Okay, then the next one is urinary system. Your urinary system filters your plasma. That's the only thing it filters. Your urinary system filters the plasma of your body, and then you can look at the textbook or wherever you have. It says a whole bunch about the urinary system. But quickly. The urinary system is made up of 2 kidneys, you guys, and then you have 2 ureters, and the 2 ureters end up in bladder, urinary bladder, and then from urinary bladder it goes to urethra, from urethra out in the environment. Why? Looking at that diagram, why do you think female have more urinary infection than male? Very good, because urethra is shorter in females, so the bacteria can travel a shorter distance and reach into the, God forbid, bladder and eventually into the kidney. Okay, so that's why female have more. I didn't say male do not ever get urinary infection. They do get urinary infection. They do get urinary infection. However, they get them less than women because the bacteria has to travel a longer distance, urethra. They have to travel a longer distance in order to reach bladder. Okay, that's why it's very important when you wipe yourself backward. So you never want to wipe yourself forward in order to bring the bacteria from your digestive tract into urinary tract. I hope I'm making some sense. Okay, so enough of that. You have a kidney, 2 of them. In this diagram, you have 1. So blood goes to the renal artery, and the blood comes out of kidney through renal vein. From renal vein, where does it end up? Inferior vena cava. Okay, we talked about all of that. Then your kidney has 2 layers. They call it the cortex layer and the medulla layer. Right here is, the cortex layer is right up here, which you will see Bowman's capsule and glomerulus. And the medulla layer, which is this portion of the kidney, and I will talk and I'll show them to you in a minute. So right here on this model, quickly, I'm going to go over and show you guys everything is going to be on the model, and then we flash through. Oh, I dropped my glomerulus. Same thing in the lab. I dropped my, can I leave it here for now, please? You are so generous. Okay, so right here, this layer of kidney, guys, I hope you can see it. You all can see it. This layer of kidney with the white stuff in here, you all know what I'm talking about. Okay, that layer right here is the glomerulus. They call it the cortex layer of kidney. So in your slides, in the lab, I just wrote another question, what layer? So I might put a piece of tape here, or on microscopic slides, I put a pointer here, what layer? That's a cortex layer. And then on the bottom, right here, that's a medulla layer. They call it medulla layer. So right here, all of the stuff right here is medulla, the parent, but I don't expect you to know that term. Okay, then if you enlarge right here, and then of course, I will talk about this. This is renal artery, renal vein, right? Red, blue. And then on top, they cut off just a chunk of this and show it to you here. Same as your small intestine in the lab. You asked me that question. So this is the cortex layer, medulla layer. In the cortex layer, you have glomerulus. Okay, all of these balls are glomerulus. Of course, it's Bowman's capsule, but inside of it is glomerulus. And then, that's all I have to say about that one. Then I come back to it in a minute. So what happens, guys? What happens? Blood from renal artery comes into the kidney. And what happens? It branches to what? Afferent arterioles, okay? Here, for this arteriole, we have a specific name. It's called afferent, A-F-F-E-R-E-N-T, afferent arteriole. So afferent arteriole from renal artery, it branches to afferent arteriole. Afferent arteriole, then blood goes into glomerulus. From afferent arteriole, blood goes to glomerulus. Glomerulus, what it is, it's a bed of capillaries. So we still follow the rules of aorta, artery, arterioles, capillary, and after capillary, where blood's supposed to go?
Venules.
To the very good venules. But this one time, this organ of your body does not follow that rule. It goes from a capillary, which we call it glomerulus, into another venule. This venule, we call it exit venule, or efferent, with E. Arrive, efferent, efferent, with E. Exit, venule. Arteriole, sorry about that. Arteriole. Afferent arteriole, glomerulus, efferent arteriole. And from here, then the blood goes into capillary, from capillary to venules, from venules to renal vein, from renal vein to inferior vena cava. Do I make sense so far? Okay, so that was the passage of the blood. One of the questions at the beginning, what is the passage of blood in the kidney? That's it. I just said it. Okay, quickly, let's recap one more time. So blood goes from renal aorta to renal artery, from renal artery to afferent arteriole, to glomerulus, to efferent arteriole, to capillaries, right here. The capillaries for that would be here. And then from capillary, it would go to venules, from venules to renal vein, from renal vein to inferior vena cava, and into the right side of the heart, right atrium. You remember that? I'm finishing the cycle for you guys. Blood cycle. Okay. Then what happens when blood comes here, you guys, in the glomerulus, right here, you do have slide of it in the lab. So this is glomerulus, a bed of capillary, just like a capillary, just like a cord. If I had a cord, I would fold it back and forth. It's not a regular shape, it's a regular back and, folded irregular shape. That's what glomerulus means. Glomerulus means fist. Okay, so that's what, in the glomerulus, when blood is percolating, like a percolator, you have coffee percolator, whatever it is, Then you have outside of it surrounded by Bowman's capsule. This is called Bowman's capsule. This right here. So the pressure in Bowman's capsule is less than the pressure in the glomerulus. And what happens, small molecules, not large molecules, small 6 angstrom or so, somewhere in that neighborhood, they seep out from glomerulus to Bowman's capsule. And of course, blood is circulating, and that's where plasma is being filtered. If you see blood in your urine, Houston, we've got a problem. That's right, the blood does not pass through here. It's not supposed to pass through here. And of course, the blood goes through here, then end up into urethra and in your toilet, you see blood. Oh, God, I have blood in my urine. You go see a doctor, not me. If only cops knock my door, I say I'm a doctor, you go away, right? That happened. I took my child from the hospital. I took her home. I said I told my wife they're gonna kill her here if we leave her here. And the cops came at 3 a.m. Where's the baby? Are you a doctor? I said, yes, I'm a doctor. Are you a medical doctor? I said, no, I'm not a medical doctor, but I know more than those doctors in the ER. They're going to kill my baby. Oh, really? No, really? But anyway, long story. I'll tell you guys something.
Now I'm curious.
I'll tell you in the lab when we have time, right? But, so when small molecule seeps out from glomerulus to Bowman's capsule, then the filtrate, that's called filtrate. I wish I could, yeah, it's called filtrate. So right here, it's called filtrate. Filtrate is plasma. What it is plasma minus, my sign minus. You all know what minus is. Plasma minus small molecules. Okay, so that's pretty much what is in here. So small molecule seeps out into the filtrate, and from filtrate, then it goes to proximal convoluted tubule. I know it's tough. Here's a glomerulus. Here's a proximal convoluted. It's a convoluted tubule. Proximal convoluted tubule. Then to loop of Henle. From loop of Henle, filtrate. From loop of Henle, it goes back to distal convoluted tubule. And from distal convoluted tubule to collecting duct. From collecting duct, it goes to minor calyx right here. And from minor calyx to major calyx, 2 or 3 minor calyx are combined together. It makes up major calyx. We only have 3, human have 3 or 4, 2 or 3, 4 major calyx. And from major calyx goes to renal pelvis, from renal pelvis to ureter, from ureter to bladder, from bladder to urethra, and out into the toilet. Right? Am I making some sense? So I gave you the passage of filtrate, how it's being filtered. The plasma is being filtered. Here it is. So the pressure, quickly, I'm going to let's talk about this. The pressure in the glomerulus is greater than the pressure in Bowman's capsule. So what happens? Small molecule seeps out. When the small molecule seeps out, they go to proximal convoluted tubules. From proximal convoluted tubule, and here it is, this is the glomerulus, Bowman's capsule, and all of these are your proximal convoluted tubule and distal convoluted tubules. Which one is which in this slide or any slide? I don't know. I don't know if anybody can tell which one is which. I mean, proximal convoluted tubule and distal convoluted tubule. I don't know. But I know this is simple squamous epithelial tissue, and these are cuboidal. All of your convoluted tubules are cuboidal, simple cuboidal epithelial tissue. The glomerulus is simple squamous epithelial tissue. I hope I'm making some sense. So that's the what layer? It's the cortex layer. Because of the glomerulus. I see the glomerulus here, I call this the cortex layer. If I don't see glomerulus, then I do not call it cortex layer. Look at this cuboidal epithelial tissue. Nice, beautiful cuboidal epithelial tissue all around it. And this is the lumen of the convoluted tubules. Okay, here's a scanning electron micrograph of, and here is, quickly, I took it from physiology class, whatever it is. Here's the glomerulus, here's the afferent arteriole, glomerulus, efferent arteriole, Bowman's capsule, and then proximal convoluted tubule. Why proximal? Is in the close to this Bowman's capsule, so they call it proximity. And then this, they call this distal convoluted tubule, is distance away. Nick is proximal to me, and then wisdom is distal from me. Right? I hope I'm making some sense. Proximal and distal. That's what they call it, proximal to glomerulus and distal to glomerulus. And then the filter goes through the loop of Henle, from loop of Henle to distal convoluted tubule to collecting duct and out. Here it is. Oh, this one, what they, you will see this term later on in the semester, countercurrent multiplier. What happens here when the filtrate is going through the loop of Henle, right here, a few things happen. Water goes out, and then sodium, salt goes out, and urea comes in. A few other things happen. That's what they call it, counter-current multiplier. When the current is this way, the molecule are entering this way. So do not worry about it for this class. But when we get to clams, when we talk about clams later on, that term shows up again. I thought I might as well throw it at you here. Don't worry about it too much. They spend in physiology, they spend half a day or so talking about it, what is current multiplier. But not here, we don't worry about it. Okay, I'm done with urinary system. Quickly, we cover these things to go over, finish it up. Digestive system, again, I'm not too concerned about. You worry about pharynx and soft palate, tongue, so on and so forth. Just epiglottis, make sure you know epiglottis. It's a flap of cartilage, if you would. It's made up of cartilage. And it closes, when you're eating, it closes the passageway to your trachea. So food does not go to your trachea. It is interesting. In my culture, where I grew up, during dinner time or lunchtime or breakfast, they said, do not talk while you're eating. And I came to the United States, and in the United States, I go to American families when I came. I said, how come you don't talk? And they were eating. I said, am I supposed to talk? I said, no, we talk during while we're eating. So I don't know. I didn't choke so far in my life. I didn't die.
Don't tempt it.
I don't know what, but again, now I understand both cultures. Yes, I understand it now. Before I was confused. But anyhow. So the flap of cartilage goes here. So you're not supposed to be talking, laughing, I don't know, whatever it is. And if food goes into wrong pipe, they call it, goes to trachea. Houston, we really got okay. And then the food from your mouth is supposed to go to your esophagus right here. Okay? So that's trachea. And don't worry about the larynx, the voice box and all that. Okay, I'm not too crazy about that. So what happens, soft palate, again, you guys, I'm not worried about soft palate and hard palate, all that. The food bolus goes to your esophagus. In the lab, we do have slide of esophagus. And when the flap closes, here we go, food goes to esophagus. I'm waiting for the term propel towards stomach. Contraction of esophageal muscles propels food to the stomach. Do I have it? Yes, peristalsis is right here. Okay, so let's, I've got to get used to these notes. Okay, so digestion, gut movement causes segmentation when you have, and I will talk about that, alternate constriction of rings of muscles to divide gut into contents, mixed food, but does not move it through gut. Peristalsis, wave action of the contractions, which I will emphasize on that. I will talk about peristalsis, but I will not talk about segmentation, and hopefully I will talk about both of them, where your gut occur. And then contraction, when the inner circular muscles contract, here we go. I gave you the definition of peristalsis. When this inner circular muscles, so you have two layers of muscles in your esophagus, and also in your small intestine, not in your stomach. Your stomach have three layers of muscles. If you look at the slide of muscles, you have the circular, longitudinal, and oblique. It's in an angle. But let's stick with esophagus for now. So in esophagus, you have ring muscles like this, and then you have longitudinal muscles outside of it. And food is right. You all follow what I'm talking about. So you have food right here, which is called lumen. Anytime, anywhere in your body, you have a tube. Inside of that tube is called lumen, L-U-M-E-N. It's supposed to be hollow and material pass through, either blood or filtrate. We just talked about that, or in this case, food. Okay, so you have circular muscles here and longitudinal muscles here. So it would be like this. It's sitting in my esophagus just like this. So longitudinal muscle this way, my fingers this way, and circular muscle this way. So what this one is saying, when peristalsis occur, when the peristalsis, both of these contract and relax, not at the same time, thank God. What happens when the inner circular muscles contract, the tube tightens. So when these are smooth muscles now. Remember, you have three types of muscles: cardiac, skeletal, smooth. You remember that? This is a smooth muscle. When you swallow something, it's gone. You cannot bring it back up. I will not say how, what. I've never done it in my life. Because I don't know how some people do that. But anyway, it's gone. You swallow 50,000 sleeping pill, right? It's gone. They have to go in there with a vacuum to take it out. Patient who commit suicide. And then what they do, they go to the hospital and they put a vacuum, they put sleeping pills out. If they don't, the person dies, right? You all know. Okay, so what happens, the circular muscles contract and then the tube tightens right there. I expect you to know that. And when the longitudinal muscles contract, the outside longitudinal muscles contract, the circular muscles relax, the tube get loosened. Okay? Get loosened. And then, right here, they're showing you peristalsis here, and peristalsis occurs since your textbook talks about it, I will go ahead and elaborate on it a little bit. Peristalsis occur in your esophagus only. When food goes to the small intestine, you have three segments of the small intestine: duodenum, jejunum, ileum, in that order. Duodenum, jejunum, ileum, in that order. Okay, in the lab, you don't have to identify them separately, but there are structures in there I would like you to know in the lab. So in the small intestine, then you do have segmentation and peristalsis. So in the small intestine, you have segmentation and peristalsis. Right here, here is a model. The camera, Ruben, does the camera take the model here? If I go over the model here, will it take it?
No.
No? Okay, I'll move it over. Don't worry. Okay, so let's go over. Here is small cross-section of small intestine you have in the lab. These are villus, scan electron micrograph of it. These are villus, the finger-like projections. I will show it to you in a minute. And this is a lumen. You remember I talked about lumen? Any tube you have inside of your body, any tube, blood vessel, whatever you have that inside of it is supposed to, is hollow and supposed to material goes by, it's called lumen. And then of course you have your muscular layer, which are two layers of circular and longitudinal. Okay? So here's a cross section. of the small intestine, and that's what you have in the lab. And these are villus. Villi is plural, villus is singular. So inside of villus, you have capillaries right here, and then the cells, you know, I'm going to show it to you in the model here in a minute. The cells on the villus are, right here, the cells on the villus are simple columnar epithelial tissue. Are you with me? So, here they are. Here is villus, here is villus, here is villus. Together, they're all villi. US singular, I at the end, plural. Okay? So, what happens, and inside of villus is capillaries, right, to absorb food and nutrient. And then there is that green stuff, it's called lacteal. Lacteal is for absorption of large fat molecules in your food, the food that you eat. And then these cells right here, you see some blue ones in there. The blue ones are goblet cells, but these cells are, if I put a piece of tape here, ask you a tissue that is simple columnar epithelial tissue. And if you look on the diagram here, the cell membrane is folded back and forth. Back, it's not cilia. This is not cilia. Do not confuse it with cilia you learned in Bio 1. The cells here are folded back and forth. The cell membrane is folded, not the cell. The cell membrane is folded back and forth. And that's what they call it microvilli. Microvilli. It's not listed in here. Maybe next slide. Not here. Okay. It's called microvilli or brush border. Another name for it is called brush border or microvilli. Maybe I should write it down. Brush border, same as microvilli. Remember, this is villus, this entire thing is villi. Okay, so what happens? Right here, they're showing you what it is one by one, and then the layers for the lab purposes. Yes, you should know the name of these layers, guys, where you have